Dear participants, I am so glad to start our second session. It will be policy formation and global practices of One Health of our today's conference, One Health Social Dimensions. And first of all, excuse me for our timing that we need to start later due to some technical problems. But due to these problems, I am so glad to present our first speaker of the session, Eric Bortz, Associate Professor of the Department of Biological Sciences, University of Alaska Anchorage, with his presentation, Benefits of Genomic Sequencing of Swine Viruses in Ukraine, African Swine Fever, Porcine Circovirus Type 2, and co-infection. Thank you so much, Eric, for your video and for your joining to us, and the microphone is yours, please.
Okay, thank you very much, Marina. I'm very excited to be part of this conference. I'll give my presentation here in brief presentation in English, if I can. Okay, it's not sharing. Stand by there. Can I send you? Can you screen share for me? Okay, let me just send it to you because I don't think it'll let me do it from.
Eric, I will write in chat my email. It will be directly to me, and I will share immediately.
Okay, thank you. Sorry. Okay, chat.
Yes.
Marina Galat at Gmail. Okay. Okay, go. It's attaching.
Okay.
Sorry, I'm in the United Kingdom and the internet is a little bit slow in Britain for some reason.
We understand you very well because we also had such type of inconvenience, especially in our colleagues who are staying now in Ukraine and also in Kyiv and in other places, so we also could understand. And I am waiting my email. Yes, so I'm still waiting.
It's almost there. Just be a minute. I guess I could just introduce it just for a second.
Good.
Professor from the University of Alaska, and I worked with a number of really great colleagues in Ukraine from 2016 to 2020 and into 2022 on One Health in the swine population as well as in birds and poultry. So those are African swine fever and avian influenza, and then we also worked with a good number of colleagues in both on human diseases, particularly influenza, and a little bit of support on projects in other human diseases, tuberculosis, HIV, and COVID-19 in Ukraine. So this talk will be particularly focused on the African swine fever tracking and the idea that we use this new sequencing technology to track the virus, the genome of the virus from samples from outbreaks from dead pigs, and then track around to understand the epidemic patterns, how the virus was being spread. And the key was to build the technology and the expertise and have lots and lots of researchers from Ukraine, as well as some other neighboring countries, Poland and Georgia, also sent researchers to learn how to use the technology, to learn how to do the computational analysis of both human and animal importance. So this capacity building project was supported, funded by the U.S. Department of Defense. The Defense Threat Reduction Agency was the principal funder. But we also had support from the United States National Institutes of Health, as well as the Food Safety, Consumer Protection, and the research institutes in Ukraine that put in a lot of time, effort, and personnel into those projects. So I'll just give a brief presentation of some of the results and some of the ideas of building that capacity here. Okay, can you share it, Marina? Did that work?
No letters yet. Just now check. Maybe you will try again to send me.
Yeah. So, Marina.
Eric, you can try to share your screen actually, because I think now you have the function to do that.
No, I think it's the UK has a firewall that won't let me do it.
Oh, okay.
Yeah.
Oh, Eric, I received, but I think it was yes, I could open.
Okay.
It was received just in the last few seconds.
Yeah, the internet's really strange these days. It's just the security.
So now it is opening. Great. So I'm looking for that I could show to all participants.
And it's great to see as many of the names here I've seen before, a few of them, and then other people we work with.
Yes.
Okay.
Good.
Should I start? Do you want to do slideshow?
Yes. And let me know if I need to do it.
To the next. Okay. Thank you. Okay. So could you go back one, please, to the title, just so we can see? Yeah, so thank you. Thank you, Marina. So the idea is to highlight the benefits of genomic sequencing of swine viruses in Ukraine, and African swine fever was our key pathogen of interest in this because of the widespread economic impact of that outbreak. And it's still going on, of course. The tracking of it has become obviously difficult, but African swine fever is still there. There's not a good vaccine. It relies on diagnostic testing, but in the veterinary service in Ukraine has done a lot of work to try to be able to get samples efficiently to the laboratory in Kyiv to be able to trace the virus. But we brought in this genome sequencing technology to build on top of that. Okay, so the next, please. Thank you. So I won't really go too much into the pathology of African swine fever, other than to say that it is a lethal hemorrhagic disease of swine. There have been massive outbreaks in Russia, China, other parts of the world, Poland, and of course Ukraine. The incubation period is pretty long, but death usually follows that. In the pigs, they have a lot of hemorrhagic disease. The pathology is pretty interesting, but you end up having to cull and have protection zones around the affected farms, as you all know, because you guys built these safety measures into outbreak investigation. But that's the only real measures you can do at this point. Once you have an outbreak, there are vaccine candidates, and people are talking about them, the USDA and so on, but those are not ready for manufacture at this point, and there are no antiviral drugs. Next, please. So diagnostics are the key. and biosecurity, other tools that you use for One Health to track ASF in Ukraine and understand the epidemic patterns is this new genome sequencing technology. As of to date, there's been over 546 recorded ASF outbreaks in Ukraine. It's actually a bit more than that now. So, and that's from 2012 to 2022. So 10 years of major ASF. Next, please. And if you look at the affected commercial farms, backyard farms, there's also wild boar sampling by sampling from hunters. So the surveillance system is in a lot of ways reactive in the domestic swine population. It's when there's an outbreak, then there's detection and testing. For wild boar, it's opportunistic. It's when a wild boar is, hunted or left on the side, discarded, that it can be tested. But you can see the curve goes up. Yeah, next, please. There we go. Thank you. The other thing is there's a seasonality to it in northern Ukraine. In the northern parts of Ukraine, in the forests, near the forested region, and a lot of the rural and farming areas, wild boar outbreaks are in winter, and domestic pig outbreaks are really July, August, September, so autumn. And you see this pattern, and it's pretty strong in northern Ukraine. In southern Ukraine, it's not the same. It can happen kind of all year, but you do get some more domestic outbreaks at the same time, August and September. Next, please. The other thing we measured as part of the epidemiology of this project, and a lot of this was the work of Maxim Bezymeny at the Institute for Veterinary Medicine in Kyiv. He was the lead researcher doing our GIS analysis, at least on the samples we were working with. And there's other people involved in it as well. Was to look at, showed that the further, if you mapped, further you got from the borders of Ukraine, at first the samples were dominated by wild boar, but then later on, as the epidemic progressed, there were more outbreaks in the interior of the country, away from the borders to some extent, in domestic swine herds. So we have a little bit of data trying to show that it's not necessarily statistically significant. As we see, it's just a trend, it's a pattern. Next, please. So in sum, we've observed multiple clusters of ASF outbreaks, and we're wondering about transboundary spread by wild boar and pig products. So does ASF, is it spread from distance by boar? And this is seen in Poland. It's really been studied a lot in Poland, actually, with our colleagues in Poland, and then causing these outbreaks, bringing it into one farm, into one oblast, into one region, another, and then spread amongst those domestic farms, especially the large commercial farms, but also backyard farms, which are a little bit undersampled in these situations. So next, please. So one of the ideas was that we could do maybe genotyping of the virus. So the first thing is what strain of ASF it is. Everybody knows it's the Georgia type 2 lineage. Some of the first work that we did was to sequence a little bit of the virus genome and say that it is the Georgia lineage. When you do that and you look at multiple samples from Ukraine, they all look exactly the same because they're only looking at a little bit of the virus genome. The virus genome is actually very long in DNA. 189,000 nucleotides of DNA. So if you're only looking at a little bit, they all look the same. It proves that it's genotype 2, this virulent Georgia origin genotype that came in 2007, but it doesn't let you do tracking of one outbreak versus another. So we adopted a more higher technology approach. Next, please. And that was to do genome sequencing of African swine fever. And this was really led by 3 junior scientists, at least in the laboratory, doing the actual laboratory work, doing the analysis on genome sequencing and bioinformatics. 3 scientists, 2 of them from Ukraine. Those are Mykola Sushko and Gana Kovalenko. Did a lot of the original ASF samples and sequencing on one of these portable sequencing devices and the bioinformatics analysis. And then Annelise Ducloseau is a French researcher, and she was mentoring them in this process. And the rest of us, we were, professors and helping out and figuring out what samples and trying to understand the patterns and teaching some of the methodology. Okay, so next, please. Go one more, please. Yeah, so this is called nanopore sequencing. It's a little nanopore device. It's about the size of a cell phone, and you can put DNA into this device in a library. You spend a day or 2 in the lab preparing the DNA and tagging it, and then you can sequence whatever that DNA is. It can be an environmental sample, a sample from a disease. It can be an infectious agent, a bacteria. You know, it could be basically the genome of anything. And we sequence a lot of different things on this. And it goes into this little nanopore device. It produces voltages. You interpret those. Next, please. As a nucleotide sequence through, you know, it's called, you know, base calling and bioinformatics assembly. And then you pull out the actual complete genome sequence. What good is that? Next, please. Next one, please. Is it stuck? Stuck. No, one back, please, one back. Yeah, there we go. Thank you. So if you look at that, you can read not just that one little bit of sequence of the virus, but we can read many, many, many parts of the long virus genome. And when we do that, we can put them together, which ones are the same as like a fingerprint and pattern using a phylogenetics method. And you build a little phylogenetic tree there on the left and point where, you know, where are the locations and times of those particular outbreaks. And when you look at the tree, we kind of get an inkling that, you know, we get an idea that there are outbreak clusters, at least some of the, you know, some of the genotypes of the virus, some of these little signature SNPs or single nucleotide polymorphisms are called SNPs. These little signatures are all the same here and all the same here and all the same here, indicating that maybe there was some sort of localized spread within, you know, this region. So next, please. And Mikola really, he really looked at that. We also applied that method to understand another virus, which is porcine circovirus. Go next one more, please. And by doing the same kind of method, and this was work out of Kyiv with the Institute for Experimental and Clinical Veterinary Medicine, and that was Natalia Rudova and Anton Gorilovich and Alexei Solodyankin. They were able to, and working with one of our students in Alaska, we were able to do this with another swine virus and show that there's potentially a wild boar and pig transmission event that must go on, because the virus genotypes are very similar with, you know, in both types of samples. And those samples were up from the region along the border of Kharkiv and Sumy and Chernihiv Oblast. So we think that there's this transmission event that happens with wild boar up in these forests. And so that's the same kind of thing that we saw with the African swine fever. We saw with another swine virus, giving us confidence that this method is pretty good. So one more, please. Next, please. Yeah, so then we went on to sequence the complete genome. That's fine, we can stay here. The complete genome of a number of more viruses, not just the little pieces, but the entire genome of the viruses. The first genomes that Mikola and Anna had worked on, Anna Kovalenko, she published one of these genomes in a paper, and we, working with Mikola, we took the other 11 genomes. So we have a whole bunch of them from Ukraine. We put, and she did an analysis on a phylogenetic tree to figure out, you know, the clusters through time and where those clusters potentially can be. And you do see that in 2018, there are viruses that probably went from wild boar to pig, and you can trace those in Ukraine. So this is just sort of a proof of principle to build the technology and the ability to add another dimension of information to tracing ASF outbreaks. And it can be applied to many, many veterinary diseases and human diseases for that matter. And it really through using this genome sequencing technology. Next, please. One more, next, please. Okay, so that's it. In summary, the virus genome sequencing informs the one. Could you go one back, please? One back. Yeah. The summary. Okay. Yeah. So we sequenced the full genomes of African swine fever using portable sequencing technology. We also use these little signature sites. If you just use the traditional little bit of genome that you use for diagnostics, it's not enough. You have to do longer genome sequencing to be able to do this type of method. And it's been used in COVID-19 all the time. We use this in SARS coronavirus to track human infections, HIV, and so on. You can also apply this to veterinary diseases. It appears that the epidemiological analysis that there are multiple introductions of ASF into Ukraine from other countries originally, and then spread within Ukraine. Maybe wild boar will carry a lot of it over the borders, and they're spread within the domestic pig population through farming practices and trade and so on. The other thing is this team, when you build this team to do this, you have to have multiple kinds of expertise, biologists, veterinarians, people working consumer protection, One Health, computational bioinformaticists, and so on. So you have to have a collaborative group with different institutes. And the idea is, how do you use this information to report back to the stakeholders, the farms, the government, the funders, the vet services? So that's just a quick overview of the projects that we worked on and the benefit of applying this technology. In the future, the idea is that One Health research and tracking of diseases in animals should always use this kind of technological approach, or include it. It can be other things, but it always is good to add this type of approach. And once you build the capacity to do it for one pathogen, like African swine fever, you can use it for other things, high path avian flu, other diseases in humans, and so on. Okay, so thank you everyone for your attention. One more. This is the group that has worked on this at my lab at University of Alaska, and Devin Drown at University of Alaska also. Anna Kovalenko is now in the UK, and she did a lot of work on the phylogenetics. Then we had groups in Poland, as well as some other collaborators. In Ukraine, Mykola Sushko at the SSRILD VSE in Kyiv. I'm not going to say the whole name, the State Sanitary Institute for Research in Infectious Disease Diagnostics, Laboratory Diagnostics, and Sanitary Expertise. You guys know this institute. Anyways, he worked there with a number of the senior researchers on this, as well as at IVM, Maxime Beziameni, led the GIS analysis. Our colleagues at NULES have been very supportive of this type of work, and really talking about the epidemiological analysis, as well as some of the other people that work closely with us, Roman Ditsenko, Sergei Skokharad, and Alexander Zinenko, and I know he's here actually. So the food safety and consumer protection of Ukraine was critical to supporting this project. It was funded by the U.S., mainly by the U.S. Department of Defense DITRA program, as well as the National Institutes of Health NIGMS program. And I'd be happy to take any questions, or if you'd like to contact me, please do so at my email address. Thank you. Okay.
Thank you so much, dear Eric, for your presentation.